So as I warned you, we're going to delve into model systems quite a bit, and I wanted to introduce you to the nematode C.elegans as a model that we have used very productively to define biomarkers that now we're not looking, in this case, for biomarkers of age, but of future longevity. Next slide, please. So this is the nematode C.elegans. It's a free-living, soil-dwelling nematode that at its adult size on the right is 1 millimeter in length. Do I have a pointer? It starts life as an egg and goes through 4 larval stages, L1, L2, L3, L4, before it finally makes the cover of Science magazine as an adult. It also has an alternative stage 3 called the dower, which is actually intermediate in size between L1 and L2, but it replaces this as a state of developmental arrest that seems not to age. Once they come out of dower, they can resume after a month or 2 in this holding pattern. They can resume development and become fertile adults with normal lifespan. Next slide, please. So there are many advantages of studying longevity in C.elegans. Next slide. You have to keep hitting so I'll just say next, and it'll be the next one. So it has a short lifespan, normally about 3 weeks or a bit less, so you can do very quick assays, and it can be semi-automated. Next. You can actually functionally test the role of any gene that you suspect affects lifespan by a very easy process of knockdown where you in fact just feed bacteria expressing the particular RNA interference construct. Next. It's very easy also, fairly easy also, to introduce transgenes to begin to express something that was not made. Next. And we already have a lot of mutants and reporter constructs to help us with the analysis. Next. And there's a huge power for gene mapping, even for something that is complex with many, many genes contributing to the traits such as longevity. And this is important because longevity is a difficult trait because of its multigenic nature. Next. So why develop longevity biomarkers in C.elegans? Next. First of all, we didn't know it at the beginning, but we now know that many of the longevity pathways, many pathways of all sorts, including those that affect lifespan, discovered in C.elegans are conserved in humans. This includes pathways for apoptosis, but also insulin-like signaling, nutrient sensing such as mTOR, AMPK, etc. So it's a good model for conducting initial surveys. Next. And because of the reverse genetics, we can test the causation of factors that we suspect. And a good marker of longevity is, the better the marker, the more likely you are to be close to the proximal cause. Next. And of course, if you get good biomarkers, then you can use these to the extent that they extrapolate to humans. You can use these as surrogates for actually waiting until death to determine if an intervention really affects the rate of aging or subsequent longevity. Next. So we began with a panel of C.elegans longevity mutants that we knew by published reports differed in lifespan by at least a factor of 2.5. And we did what geneticists are inclined to do, which is to outcross these into a constant wild-type genetic background, so that essentially the only thing that differs between them is the mutant that we know about, because otherwise there would be other background differences. And what we found was certainly a variation in lifespan, going out to almost tenfold the normal lifespan. This is the same double lifespan mutant that Cynthia Kenyon discovered that's out here in this graph. So This is now compressed in the lower panel. Next, please. And next. And some of the mutants were disappointing. When we studied them in a constant genetic background, mutants that had been reported to be long-lived against admittedly different controls turn out not necessarily to be very long-lived. So some were disappointing. Next. And a couple, 2 mutants in the PI3 kinase catalytic subunit, age 1, were better than advertised. They were reported to live 2 to 2.5 times the normal lifespan. And in this experiment, it was almost 9 times. In another experiment, it was 9 to 11 times. So they're roughly 10-fold longer lived than the wild type when you knock out a gene. That's a surprising thing, that you can improve performance when you remove functionality for something that should be helping the organism. But the secret is, of course, that the main function of any of these genes that, when they are knocked out or knocked down, increase longevity, is that's not their main function. They have another function, typically to do with reproduction or development, and when you eliminate that, they may be very slow developing or even fail to reproduce, but they live a long time. Next. So these very long-lived mutants, the 10-fold long-lived age 1 mutants that are deficient in PI3K, can't complete larval development at 25.5 degrees, the high temperature that many labs grow them at. But at a lower temperature, they slowly complete development. It takes them 4 to 6, maybe 8 times even as long to develop as normal worms. But when they do, they're very long-lived. Next. And if you'll click on this, you can see at 8 times the median lifespan, median not found, it's not going to work. You'll have to take my word for it. It moves around just like a worm, a wild-type worm, 1/10 of its age. I was afraid of that. Just go on to the next slide, it's all right. We had live video there, but it's not working. So this is the diagram of the domains in this PI3 kinase catalytic subunit. There's a binding site for the regulatory subunit, a RAS binding site, a C2 domain, a helical domain, and the kinase domain. This is the business end, the functional part of it that, in fact, is responsible for lipid kinase activity. And the most commonly used mutant discovered by Michael Klass and championed by Tom Johnson, has a mutation that we actually were the first to map that is an extension of the helical domain, presumably into the kinase domain. So it loses quite a bit of function and is about, at best, 1.5-fold long-lived. The 2 different very long-lived mutants are both stop codons, so they're nonsense mutants that makes the protein synthesis terminate upstream of the kinase domain, so they have no kinase activity at all. Next. And they're very long-lived. Next. So what is this PI3 kinase doing? Well, it makes a product called phosphatidylinositol triphosphate, or PIP3 for short, out of PIP2, and it's opposed by a phosphatase, P10, which incidentally is also a tumor suppressor gene. So it tells you that the amount of PIP3 that's made has to be controlled, and if you don't control it, you tend to get cancer. Next, keep bringing these up. So this is age 1 making a little bit of PIP3. Maybe only 1 part in 1,000 of PIP2 is converted to PIP3, except under stress, in which case that percentage can go up 10 to 100-fold. And the phosphatase quite effectively reverses that reaction. So the product, PIP3, sits in the membrane where it has 2 lipid chains, shown here as R1 and R2. Those sit embedded in the inner cell membrane, and the inositol head group sticks into the cytoplasm, where it's basically a docking site for a lot of different proteins. There were, at last count, over 35 different classes of proteins that had been found that bind to PIP3. The most common are called plextrin homology domain proteins, and for simplicity, I'll call them all that, but there actually are many different families. And the benefit of them docking to the inner membrane is it reduces the effective space that they occupy from 3 dimensions, the total inside volume of a cell, to 2 dimensions, just the inside surface of the cell membrane, where collisions are more likely. So it's kind of like the electron transport chain is clustered on the inner membrane of the mitochondrion. You have to have things brought together. Next. So when we looked at the stress responses, they were kind of interesting. Let me tell you first of all that the very long-lived mutants are not particularly heat shock resistant. They do a little bit better at the longest time, but they're actually a little sensitive to heat. But they're very resistant to hydrogen peroxide. So this is wild type in red. In green is the weaker H1 mutant, and the DAF2 mutant is about the same place as well. That's an insulin receptor ortholog. And these 2 very long-lived null mutants are much more resistant. Same for paraquat, another oxidative stress, or for H&E, which can be regarded as an oxidative stress, but it's also an electrophilic stress. So these worms are very stress resistant. Next. So this is just to show that if you knock it out, you really don't see the red stain that is now showing the antibody reaction to the PI3K, and it's now reduced to roughly the level of background. Next slide. Much more importantly, its product, PIP3, is now reduced strikingly. So, I mean, it wouldn't necessarily be true if there were other routes to make PIP3, so we had to be sure that it was knocked down. And so here is the wild type level, here's the weak mutant. This is the first generation homozygote at day 8, and this is the second generation, and this is a control with no primary antibody at all on the right. So basically, again, you're getting down to background levels, and even the first generation homozygotes that have the same genotype as these have quite a bit of PIP3, and I'll show you in a minute why they still have some. Next. So I will talk a little bit about first generation and second generation H1 null homozygotes. And the reason is because these 2 generations are stunningly different in their phenotypes. The first generation, well, let me show you first what we mean by these generations. So the color here indicates genotype with respect to H1. So there are, in these mixed cultures, there are some wild type, there are some completely devoid of the H1 functional allele, and there are some heterozygotes that have 1 copy of each. Next. And their progeny, the heterozygotes progeny the heterozygotes will lay eggs that are genetically of those 4 types as well. Next. But now below the line, here above the line we're looking at the DNA, what's in the genes. Below the line we're looking at what's in the cells, in the phenotype, how much PIP3 is there. And now it's the same intermediate amount for the heterozygotes, but they're able to transfer some of the PIP3 or the PI3K messenger RNA or something upstream of that to their eggs, which next then gets to, when develops, is diluted enormously. So there's effectively hardly any of the PIP3 in the F1 adults when they reach adulthood. Next. And their eggs, the F2 generation, start life with none at all, essentially none. So this is why F2, we think, is so different. They develop much more slowly than the F1 generation, and they're infertile, but they live 10 times as long, and actually 4 times as long as their own parents. Next. So one of the things that we first noticed when looking for some important functional changes is that the kinase activity for proteins substrates, endogenous protein substrates, is pretty much gone in even the F1 generation and absolutely gone by the F2 generation, maybe a little trace of it. And everything else is a control here. So this is getting down to 8 or 7%, 8 or 6% of the normal level. And if you knock out the downstream transcription factor, a FOXO gene, you can restore this. Next slide. So you're going to hear a lot of nexts here. This is a description of what normally happens on the left here. I hope you can see the pointer, but on the left you have the insulin receptor, which is here called DAF2, next. And it, in fact, is responsible for activating by phosphorylation the Ag1 PI3 kinase. But if that's knocked out, next, then you don't make PIP3, and the PIP3 then is not able, next, to activate PDK1 or AKT, which are more downstream parts of a kinase cascade responsible for activating they're responsible for inactivating this transcription factor, DAF16 FOXO, next. And when you don't activate it, next. When you don't activate it, when you don't phosphorylate it, it is no longer sequestered outside the nucleus. It's able to enter. If anybody has a pointer that actually works, I'm sure you could see better what I'm pointing at, because this poor thing is so feeble I can barely see it. I'm sorry about that. And when that happens, there are many protective genes that are now produced under the control of DAF16 to a much higher level, including glutathione transferases, superoxide dismutase 3, CAT2, and PepsiK. So, and you'll hear more about PepsiK in the next talk, where Anjay will talk about its importance in the extreme longevity of dwarf mice. Next. So what we found that was unexpected and interesting is that when you now activate DAF16, in addition to its positive effect on these protective genes, it also exerts a feedback inhibition. Transcription factors can activate or inactivate, and here it's acting as an inhibitor of expression of almost all of the genes upstream of this transcription factor. So it's a feedback loop. It's in fact inactivating the things that would have led to its activation. Next. Next, please. So here's the model that we come up with, that you normally have a choice of two modes, that this is kind of this feedback loop is a flip-flop circuit to distinguish or alternate between these two modes. So the reproductive mode on the left has high activity of the insulin receptor, which inhibits DAF16, so it has little activity. And through transcriptional loss of transcriptional co-repressors and the addition of transcriptional co-activators, you can promote the activity of DAF16 so that it now, this is totally dead now, so that it now gives feedback inhibition to knock down this insulin receptor. And so you have a state now where DAF16 dominates, but you'll notice that it's a balance, and you can reverse this by changing these other cofactors, and you can flip back and forth between a longevity mode and a reproduction mode. And that's what normally happens in the lifespan of C.elegans. When it is in favorable environments, it wants to be in reproductive mode and produce as many progeny as possible. And when food is scarce, or when it's overcrowded, it goes into longevity mode, where in fact it now is making more DAF16, more of the protective gene products, and it's not as good at proliferating, in fact delays reproduction. Next. What we find in these very long-lived mutants is kind of an extreme case of the longevity mode, where now DAF16 has totally silenced insulin receptor. There's no PIP3 made, and you get a lot of survival genes. Next. So instead of having a seesaw that you can flip back and forth, next, you have a very heavy weight on one end, and you're stuck in this mode. So these worms cannot reproduce, but they live very long. Next. So this slide is just to show that many pathways intersect at DAF16, including not just the insulin-like signaling, but also the P38 MAPK pathway, the TOR pathway, which is a nutrient-sensing pathway, the stress response pathways, junk MAP kinase, really P38 MAPK is as well, and the RAS MAPK pathway, which is involved in, well, both of these are involved in stress response and innate immunity, but especially the RAS MAPK. Next slide, please. So we wanted to learn what genes are altered in the very long-lived mutant to get an idea of what mechanisms it might use. And this is important because to just recapitulate the same thing, to knock down PI3 kinase in mammals is a very risky proposition. We depend on it for cell division. So next slide. The idea was also that if we learn what genes are affected in the longest lived worms, then these might also then be studied in intermediates, mutants that have intermediate longevity. Very interesting initial observation was that this is kind of a heat map where green means that you have less expression in the long-lived worm, and red means you have more. And in the comparison between the very long-lived strain and the weak mutant, and also if we do the comparison between the very long-lived strain and its parental generation, you see mostly suppression. So most of the genes that are differentially expressed are downregulated by a factor of 12. Whereas if you compare the very long-lived mutant to wild type, it's still 60-40. It still favors downregulation and more heavily downregulated genes, but it's more of a balance. Next slide. So starting with those kind of lead mutants in the very long-lived mutant, lead genes, we filled in the gaps in between. So what you see here is relative strain longevity. This is the very long-lived mutant. This is a DAF2, DAF12 double mutant, where it's defective both in the insulin receptor gene and also in a nuclear hormone receptor, and it's about fourfold long-lived. And these are some other mutants in various other pathways. And this actually turns out to be a very nice method because we found this NSPD2 gene that has a correlation coefficient with longevity of 0.99, a protein kinase D homolog that has a correlation of minus 0.98, and PepsiK, again, as I said, already shown to be pro-longevity in mice, both in the dwarf mice, but also if you make a transgenic overexpressor of PepsiK, they're called super mice. They get bigger and look fitter, but they actually live longer. And that has a correlation of 0.95. So I think this is a very productive hypothesis generator. This is a way of finding suspects, candidates that might actually be very good candidate causal factors, but also very good biomarkers for lifespan. Next slide. Oh, I was going to tell you what these are. NSPD2 is in what's called an expression mountain 4 that's age-regulated, and it correlates with a bunch of major sperm protein genes, a glutathionylase transferase, and an SOD. Next, a superoxide dismutase. This is, as I said, a protein kinase D kinase orthologous to the human one that's required for innate immunity. And this one is PepsiK, which next we already talked about it. It's important in mouse longevity. Next slide. It of course occurred to us that since the longest-lived mutants are aging more slowly. That just, if you look at just a single time point, you may be reflecting the fact that the worms you look at for these are younger physiologically than these. But in fact, at any time point, their expression is different from the wild type. So these are huge differences. This is another gene that's like over 1,000-fold upregulated in the very long-lived mutants at any time, and PepsiK. Next, please. Now, I just want to talk a little bit about something complex, which is the analysis of microarray data. Microarrays are a very high throughput tool to generate a lot of data. Proteomics is starting to generate comparable, but somewhat less amounts of data. Metabolomics still difficult to cope with, even though somewhat less complex. And scientists have been initially overwhelmed by the bulk of the data that are produced. And in an effort to make a simple message out of a complex set of data, a common procedure is to do what's called pathway analysis or GO analysis, gene ontology analysis. And it typically throws away everything that you learned about expression of these genes except one bit of information, that they're differentially expressed in your experimental group. So the only question that's asked is the fraction of genes in this GO term that is differentially expressed higher than the fraction in the rest, or phrased another way, among the differentially expressed genes, are there more of this GO term? Next slide. So what we developed, Bob DeLongchamp and I developed with Taiwan Li, is a method of using more of the information. And so instead of just looking at whether a gene is in the GO term or not, we actually took the p-values for their differential expression, which is, in a way, it's a statement of how much they're overexpressed in meaningfulness rather than in degree. So how much is one group significantly different from another? And you can aggregate these p-values. Now, if they were totally independent, if you've had a little course in statistics, you would know that if they're independent, you would multiply the p-values. But we have no reason to assume they're independent. In fact, very often, the terms within a gene ontology category are co-expressed, so we have to correct for the coefficient of covariation. And when we correct for that, we get an adjusted aggregate p-value for the whole category. This is a much more powerful method than the routine method of GO analysis. Next slide. And so, whereas conventional analysis showed only a total of 12 GO terms that were highly significant in these analyses between the very long-lived worms and wild type, we got well over 100 such categories and literally over 1,000 genes that participated in those when we did the aggregated GO analysis. So this is a much more efficient method of analyzing the data for meaningful patterns. It's both more sensitive and more specific. Next. Next, next. Next slide. Okay. So the genes fall into several different patterns. Some of them are proportional to, in their expression to longevity. Next. About half of them are like that. About a third are really only differential in the longest lived strain. Next. And about 15%, they have expression change in one of the long-lived strains or the other, but it's not proportional. So these are the most interesting ones, I think, as modulators of lifespan, that you should have proportionality. It doesn't guarantee it, but it's a good way to narrow the field. Next. And when we look at that proportionality, we find that there are some really interesting categories of genes that show proportionality the best. So having the green genes in this half sector and red genes in this triangle is an indication of that kind of proportionality. And the most significant gene class was now determination of adult lifespan. In the initial Go analysis, it didn't even come up as significant. Zinc ion binding, several different metal ion bindings, in addition to this, showed up as significant. Structural constituent of cuticle, maybe not so interesting, has to do with molting. Transport and many subcategories of transport, protein, amino acid phosphorylation. These were ones that turned up as proportional. But there are many more that, if you just group them all together, show a lot of green sector points down here and red sector points here. Next. I can go into more detail about that at some other time if you like. Changes in metabolism were long suspected to underlie greater longevity. Maybe to be long-lived, you have to slow down metabolism. Well, it turns out that the very long-lived worms are not low in metabolism. They actually generate more carbon dioxide, and they consume more oxygen by almost a factor of 2 than either the weak mutant, which is much less long-lived, or wild type. If you look at the respiratory quotient, though, which is the ratio of carbon dioxide to oxygen, then there is a drop of about 20%. And try to remember this for an hour, because Andrzej Bartka is going to show you an almost identical drop in respiratory quotient for the very long-lived Ames dwarf mice. Next slide, please. Now, the last thing I want to talk to you about is a branching into another realm of looking for biomarkers that might be useful clues as to why these very long-lived worms are long-lived, and that might apply to the intermediate stages, looking at metabolites. So we used the same comparative strategy, starting with the very long-lived worms, comparing them to wild type and the weak mutant, but we filled in some points in between. Next. And we got a lot of metabolites. Some of them were the expected energy and redox compounds that you'd expect to find in the aqueous phase. But some of the best predictors were fatty acid chains, lipids. This came as a surprise to us, and these were lipids that are found especially in membranes and lipid storage vesicles. Next slide. So this is the same kind of age plot, where this is relative longevity, or the log of it, and this is the percent of total fatty acids. And you can see that as a mutant worm strain is more long-lived, it increases the fraction of short-chain fatty acids and decreases its fraction of long-chain fatty acids. Monounsaturated fatty acids, however, go up quite strikingly with longevity. Polyunsaturated fatty acids go down. Now, you've all been hearing that PUFAs, polyunsaturated fatty acids, are important for you, especially omega-3s. I think that there's some bias in our diet that contributes to that. We take in far more omega-6 fatty acids, maybe 30-fold more than omega-3s, so anything that redresses that helps us. But if you're a worm, you're better off to have less PUFAs. The average chain length, as you'd expect from these two curves, decreases as you get longer lived, and the elongases also go down as you're longer lived. Next. So you can plot these factors on a kind of two-dimensional surface. So this is going from saturated fatty acids, monounsaturated to polyunsaturated, and short chain to long chain. And what you see in these arrows are that these are the things that are elevated in the longest lived worms. These are the things that are suppressed, so they're making less PUFAs. And the genes that make these conversions are thus implicated as candidates, and they change too in a consistent way. Next slide. Next slide. How are we doing for time? So, oh, you're going too fast. Go back. So if you look at FAT4, which is responsible for making the highest PUFAs, if you knock that down as you kind of try to emulate the longest lived worms, you get an increase in lifespan. So this is an experimental test of that hypothesis. This is now looking at a benefit to hydrogen peroxide resistance, but there's also a benefit to lifespan. this gave us about a 25% increase in lifespan. Next. Similarly, FAT6 is another fatty acid desaturase. It has a lesser effect. It's not so important in making the higher PUFAs. Next. And the elongases, if you need both ELO 1 and 2, they're somewhat redundant, but if you knock them out, you get an increase in lifespan and a little protection from superoxides. So the lesson here is that the protection from hydrogen peroxide is kind of a model of what you worry about in lipids, which is a free radical chain reaction of lipoperoxidation. And hydrogen peroxide triggers that kind of free radical chain reaction. And knocking down the desaturases that make the membranes more susceptible to peroxidation is what protects you. But the elongases are probably playing a secondary role, that they just compensate for the increase in rigidity of the membrane that you would otherwise have by getting rid of PUFAs, by making the chains shorter so that it's again a fluid membrane. Next slide. There's reason to believe that the same principles apply to humans. A study published in 2008 in Rejuvenation Research showed that the offspring of nonagenarians, people who live to at least 90, if you look at their erythrocyte membranes, they have essentially the same changes compared to age-matched controls. So they've inherited half the genes from their parents. That's why you're looking at the children of nonagenarians. If you look at nonagenarians themselves, you don't have age-matched controls because they'd be nonagenarians too. But you can look at the children of nonagenarians or centenarians, and they are also enriched for monounsaturated fatty acids, for saturated fatty acids, and they have less of the polyunsaturated fatty acids, just like our longest-lived worms. So we think that the effect is for the same reason, reducing the risk of lipid peroxidation. Next slide. Next. Okay. So we also delved into an area of proteomics that has been little explored in aging, and that is the kinome, the phosphorylation of proteins. Next slide. So if you array peptides, synthetic peptides, that are known or suspected substrates for specific kinases, this is a side view of the same slide showing these peptides arrayed here, then you incubate with the protein extract from worms of different lifespans. The kinases will phosphorylate their targets and not other peptides, so you'll get a picture of kinase activity. Next slide. And you can use antibodies next to identify those phosphorylated sites. Next slide. So the primary antibodies are then visualized with secondary antibodies. Next slide. Next, please. Okay, so if you do this, you get different spots. This is a two-color superimposition showing the ratio in a knockdown. This is just really a test case. Don't stare at this too long and hard, just to show that it works. Next slide. But these are some of the data where we have the very long-lived worms, and next you can see that quite a number of these activities are much lower in the long-lived worms than in their wild-type counterparts. Next. And that's reversed for the most part in the DAF16 revertant that also is reversed for lifespan. Next. Next. Some of these are effectively zero in this case. And next. Not all of these reverse. So some of them are independent of the transcription factor. Next. So this is just a graph of some of those results to show you that a variety of different substrates are knocked down by huge factors in the very long-lived second generation, even compared to the first generation. Next slide. So that's it. Next. I'm sorry I have to keep saying next to step through this. I thought I'd just be pushing a button, but it didn't work. So we've looked at a variety, go ahead and put them all on, just keep hitting next until everything's on there. We've looked at a variety of different classes of molecules that can provide useful and I think instructive biomarkers. Transcript abundances, lipid characteristics, and proteome features, so that we can come up with some clues as to the mechanisms of longevity increase in a mutant panel, but also so that we now can develop some interesting candidate biomarkers to look at, some of which, as I pointed out, have already been validated in other organisms, in long-lived mice or in long-lived humans. So these biomarkers often translate into mice or humans. Next slide. And lastly, I just want to leave you with a slide showing the many people who've been instrumental in doing the research, including Piotr Zimniak, who's here in the audience, but a number of graduate students and my collaborator on the methodology, Bob de Longchamp, and then a slightly different overlapping group for metabolites and for the kinase assays. I'd be glad to answer questions. Thank you for your attention. That's it. And I better put this on in case somebody asks me in Russian. Okay. Oh, it's buzzing at me.
Hi. All these lipids biomarkers, how fast in the life they will be reliable? Let's say they're good for the at birth of the animals, or when the animal become mature. How reliable these predictions will be, and when in life you can measure it actually?
Those are wonderful questions that I don't know the answer to, Andre. We have just really initiated that area with one publication a year ago, and we haven't looked in detail, and I don't think anyone else has, to my knowledge, at how these markers change during aging. In all of the experiments that we did, the question that we asked was, if you look at the same early point in life, what molecules are good predictors of future lifespan? So that we're sure we're not looking at secondary effects of aging. Once you know those, then the question you ask becomes very interesting, to see how they change with aging, but we haven't done that yet.
Very interesting. I enjoyed a lot. How much these changes in lipids are dependent on the diet?
Well, that's a very good question. The diet is very important for lipid composition in mice and humans. C.elegans has the enzymes to make all of the lipids that it needs. So it is not like us dependent on dietary polyunsaturated fatty acids or monounsaturated fatty acids. Having said that, though, you can affect that composition of the worms by altering the diet. And if we feed them a polyunsaturated fatty acid rich diet and an isocaloric diet with unsaturated fatty acids, the ones fed the polyunsaturated fatty acids live shorter. It's not strong inference because lots of things can make you live shorter, but at least we controlled for calories, and it's consistent with the intermutant comparison that we made. I think that it would be very interesting, and I have a collaboration about to begin in looking at the effect of varying the diet in humans, but we have no data yet.
Thank you for your wonderful presentation. Actually, my question refers to both your introduction and this report. And if you don't mind, I'll present myself and ask other people who ask questions also to present themselves before asking.
Good idea.
Gregory Mokin from the Institute of Chemical Physics, Russian Academy of Sciences. Okay. So, from your presentation and from previous presentations of Cinti Kenyon, You told that the results for nematode are applicable for mice and even for humans. And Cynthia told that her studies resulted in the change of her personal diet, human diet, and that she felt quite nice, younger, and more alive. Are there any other insights which can help directly to humans at this moment?
So you're asking my advice on how to change your diet?
No, I have good diet also thanks to Cynthia Kenyon. Are there any other directions of the research in worms which can help our own internal worms or our health?
Well, probably if you have a good diet already, you are already deriving all the benefit that I can tell you at the present time based on what we know from worms. It strikes me that lowering lipid intake and lowering total polyunsaturated fatty acids is a good idea. But of course you can go too low in lipid intake, but you can use monounsaturated fatty acids instead of polyunsaturated. This is getting very far out of my area of expertise, however, so I hesitate to say anything more than that.
Hello. Since people were asked to present themselves,
Please.
I'm actually presenting Dr. Vladimir Titorenko. I'm his new research associate from Concordia University, Montreal, who works on longevity and yeast. Apologize, he's not well, he's ill, he couldn't attend the meeting, so his talk will be replaced. He sends his apology. I have a question about the oxygen consumption by very long mutants. Is there any difference in size of this organism compared to the wild type species, to the wild type samples?
It's a good question. The very long-lived worms at the same age chronologically are smaller because they grow more slowly. Ultimately, at 50% of lifespan or 70% of lifespan, they're almost the same size as the wild-type worms at the same percent of lifespan. But they grow more slowly, so if you were to look at 1 week of age or 2 weeks of age, they would be smaller.
And the second question, if they are slow growing, there could be a problem with their mitochondria inheritance, motility. In some mutants which have problem with mitochondrial motility and inheritance, in yeast at least, I don't know whether it is in worm, what happens, the genes that are involved in electron chain transfer, they are upregulated, so the defects in mitochondrial motility and inheritance cause certain problems in gene expression of the respiratory chain. I don't know whether it directly impacts the oxygen consumption, but certainly based on the DNA microarray profile, the delay in the problems with mitochondrial inheritance, which could be due to changes in chronological lifespan, could affect gene expression. I don't know again about whether they directly affect metabolism, but if you look at genome profiling at metabolome, you would get an impression that there are dramatic changes, but they in fact could be due to troubles with mitochondrial inheritance. Thank you.
Well, that's a very interesting point, and I thank you for making it. There are many lines of evidence that converge on mitochondria as a critical pacemaker in longevity. And I will just point out that some of the genes that are targets of the FOXO transcription factor that is at the end of this insulin-like signaling pathway that we were calling DAF16, among its target genes that are positively regulated, that we call geroprotective genes, are the mitochondrial catalase 2, CAT2 in the worm, and a peroxisomal SOD3. Actually, I've got it the other way around. It's the CAT2 is the peroxisomal catalase, and SOD3 is a mitochondrial SOD. That these are possibly protecting mitochondria from oxidative damage. Remember that the oxygen-free radicals are mostly generated in the mitochondria, so it is logical that you would want to confer protection. Beyond that, I can say that many of the gene candidates that we found differentially expressed altering longevity are in fact mitochondrial, a disproportionate number compared to the number of total genes that are mitochondrial. So again, mitochondria came up in the Go analysis when we did it the right way. That's all I can say to address that, though. Yes?
Sean Curran from the University of Southern California. When Shoshana Gottlieb first cloned MG44 in Gary Ruvkin's lab, the thing that she noticed the most is that the maternal and zygotic null mutant was completely sterile. And so I was curious how much of the extreme longevity phenotype in MG44 is due to the fact that they don't have any progeny.
Okay, so the question is, to what extent is the phenotype of great longevity and stress resistance in the PI3K null mutants that we were studying due to their being infertile? Sean, the easy answer is that when Cynthia Kenyon made infertile mutants either by laser ablation, well, either did laser ablation, which is not a mutant, or looked at fertility mutants that were non-reproductive, she was able to extend lifespan, but only by typically 1.5-fold. So we think it's a part of the story, but it's not the major part of it. The perfect control is to feed the worms PIP3, and we can reverse the fertility effect, but it doesn't reverse the lifespan effect. So it may reverse it somewhat. We haven't finished that experiment yet, so I can't tell you completely, but they're still quite long-lived anyway.